Yeah, so hello everyone. So welcome to the first lecture of the P&S Accelerating Genomics course. So yeah, this is our first meeting. I'll try to discuss briefly what is genome analysis and why we need to accelerate it. After that, we will start going over the proposed projects one by one, and hopefully you will like a few of them. Then you can indicate your preferences, that you prefer this project or the other project. And we will meet next week again to discuss the assignment. So we will assign a single project for each student. Okay, so what is the role of this course? So as this is a projects and seminar course, so we'll try first to cover the basics of genome analysis. And as you know, the genome analysis or bioinformatics in general is not a requirement of this course. So don't worry about this new field if you don't have any details about it. So we'll cover all basics that you need to successfully complete your project and understand the computational steps of the entire pipeline and find the computational bottlenecks. Students will learn about the existing efforts for accelerating one or more of these steps. And basically, as this is a short-term project, like 3 months, so basically we will assign the step. We will tell you that we need to work on a very specific step that you can complete on time. Student will carry out a hands-on project, and this is the main goal of this course, is to give you more understanding about the fundamentals of how you practically implement one of these computational steps and then improve the computation time or the accuracy. It depends on the project. But at the end, we would like to have the benefits of teaching you these concepts rather than completing a research project or publishing a paper. So what's the key objectives that we aim to achieve in this course? So there are multiple components, and at the end, all of these will help you to improve your skills at one way or another. So we will improve your basic knowledge in genome analysis. And when I say genome analysis, don't think about the wet lab where you take a blood sample, try to analyze it or deal with the chemical molecules. But this is just the dry lab where we have computer algorithms and try to get input data, and we expect to have a very specific output out of your algorithm. We will have technical skills in genome analysis and computer architecture. Depends on the project you pick. There might be some skills required in programming or in architecture design to accelerate the very specific steps. And we hope that the course will improve your critical thinking by facing several challenges or problems throughout the semester. So step by step, we will guide you by how to find a good reference where you can read, where you can find a solution to the problem, or we will ask you just to Google it. So that hopefully will teach you how we ourselves, when we face a problem while we are doing research, how we solve it step by step and gradually. And we'll get yourself familiar with key research directions. So most of the projects we are proposing in this course are part of our own research. So once you complete something, it can be published at very good venue, or it can be part of our own project and you'll be co-authoring what we have. At the end of the semester, we will have a technical presentation of your project, and that will also give you some skills in presenting your idea or the way you implement it to a research group. Okay, so what's the key goal of this course? So this is a very interesting project or course, at least to me, and I hope it will be the same for all of you. So our goal here is to learn how to efficiently implement one of the key steps in genome analysis on portable devices. And get this, so to implement it on portable devices, you want to have it very efficient in terms of peak memory or in terms of execution time or in terms of resources in general. And for that, we would like to have here a very fast and efficient algorithms dedicated only to genome analysis or genomics. What are the requirements or the prerequisites for the course? So we don't require any prior knowledge in bioinformatics or genome analysis, as I mentioned. But definitely we need that you already complete the course Digital Design and Computer Architecture, and I hope three of you or all of you have done so. And we require a good knowledge in C programming. We don't expect a very experienced student in programming, but at least good knowledge in C would suffice for our purpose. And experience in at least one of the following is highly desirable. Could be FPGA implementation or GPU programming. And by the way, if you don't know or if you haven't programmed a GPU before, so what you wrote is mostly in C. So if you have good knowledge in C programming, then the GPU implementation would be just a matter of 1 to 2 weeks to learn the basics or some fundamentals about the architecture of GPU and how you program it. We really need your desire or your passion about completing the project. So sometimes the student face a problem, then directly ping us, I face this problem, without even searching Google or without asking a very specific person about the problem and so on. So if you have special interest in making things efficient and solving problems, then I would expect at least do some couple of search before asking a question, and you have really the desire to complete the project or do nice things. But don't worry, we have several mentors for each project, and we expect to cover most of the things required if you face any problem while you are doing your project. All right, so now the time to introduce ourselves. I move this to a later stage such that we can go over the course quickly. But this course is given by Professor Onur Mutlu and a couple of researchers at Safari Research Group at ETH. So Professor Onur Mutlu is a full prof since he joined ETH since September 2015, and he was a full prof at CMU US starting from 2009. He's currently adjunct professor at CMU as well, and he got his PhD from University of Texas, Austin. He worked at several companies in industry where he established computer architecture groups in some of these companies. And he's leading the Safari Research Group. You can find more information using this link. If you would like to contact him, this is the best way to reach him, which is his Gmail address. And the research or the research interest of our group in general, starting from computer architecture, computer systems, hardware security, bioinformatics as well, all the way to the entire computing stack, from fault tolerance, hardware, software cooperation, which is the main topic for this course, where you co-design an algorithm and computer or hardware architecture to make very efficient implementation. All right, so about me. Yeah, so I'm Mohammed Alser, and I'm working senior researcher and lecturer at Safari Research Group in ETH, Zurich. I joined the group since September 2018. I got my PhD from Bilkent University, Turkey. I worked at UCLA, TU Dresden, and Petronas before that. And my PhD was also in genome analysis, where we accelerated using hardware architectures. My PhD advisors were Can Alkan and Onur Mutlu. The thesis got several awards for the contribution we made in the thesis. If you'd like to reach me, this is my email, this is my webpage and Twitter account where I share many things about genome analysis in general or just in research. So my main research is in bioinformatics, computational genomics, metagenomics, and computer architecture. I'm especially excited about building new data structure, new algorithm, new architectures that make intelligent genome analysis a reality. And I'm going to explain what is genome analysis and what is intelligent genome analysis in a couple of few slides more. So this course is with the help of many great researchers, part of our Safari Research Group. We have Juan, who's senior researcher and lecturer. We have Damla, who's a PhD student at CMU. She's part of our group as well. We have Jeremy and John Fortuna, PhD students. you can see they have really diverse research interests that will add a value to the project we have in this course. So definitely you will have great help completing your project throughout the semester. And the Juan will be the expert in GPUs. So if you pick a project related to GPU, definitely he will be involved with us in these projects. So it will be great and helpful to go through our website, Safari Research Group, and check their profile and see what are the things they are working on. Okay, so what we expect or what we require from your side to complete this course. First, the attendance is required for all meetings. So today we arranged for this meeting for this time, but the upcoming meetings probably will be in different times based on the agreement between us and the students. We already assigned the learning materials, so we expect that you already go over them, watch at least the videos, or read the papers if you're really interested in some of the learning materials that we assign. And each student will carry out a hands-on project, as I mentioned. And give me a second. Okay. Sorry. Okay, yeah, there's a hands-on project where you will build, implement, or modify existing code, and then you will implement a few ideas where you can improve the speed up and the accuracy of the algorithm. Of course, we require participation. We will have a GLIP group. For those who don't know what's a GLIP, it's similar to WhatsApp, but we use it for research purposes. We will have several groups there or teams in GLIP. And we will have the mentors as well as each single student in a different group. So we would like to participate over there, asking questions, contribute, have ideas, or even a problem in the project itself. And this will be using it between the meetings. So we'll have weekly meetings, single meeting every week. And if you face any problem, just directly ping us on GLIP about whatever you think. At the end of the semester, we'll have presentation, and you need to deliver a GitHub repo for the project where you have all scripts, codes, implementation, and so on. And definitely, if you have a very good progress, then we will help you to get your work published. And we have a very good experience with the previous students where we are pushing for a publication at top venues based on what they have contributed during this course. So this is the main course page, and you'll find useful information. We will use your email and Moodle for all announcements, so please check them frequently. And we will assign the first, the project preferences will be through Moodle, where you can pick the few projects that you like and you'd like to work on. And after that, we can assign a single project to you based on your preferences. So about next meetings, we'll give you a chance, probably a few days, to select a project based on what we describe. And then we will have one-to-one meetings, meaning that we'll meet with each student individually to match your interest and double check that you really understand what we expect from your side. And if you like the project, we confirm that this is your project. And then it's important that you study the learning materials before our next meeting, because next meeting will be starting the project. So we don't want to spend more time on reading, but rather start directly teaching you about the very specific step that you will do. And we will start project next week, as I mentioned. All right, so do you have any question about the logistics in general for this course? Okay, so let me go quickly introducing what is genome analysis. Okay, so as you can see in this picture, if I ask you what's genome analysis, you could maybe infer that we'd like to understand why humans are different. So by comparing or identifying or do some measurement to compare some features in our genome or DNA, we hope to find these differences such that we can understand what makes this person different from the other person, for example. And this is what is genome analysis. However, unfortunately, we don't have any machine so far that can translate your blood sample or the molecule you have, could be DNA or RNA, into a text format. This text format is needed such that we can build a new algorithm or computer software that can analyze it and compare this text to another text and try to find these differences that may cause a disease or just a phenotype, which are the symptoms, for example. And regardless the machine that we use to read the DNA or the genetic material, which we call it a sequencer, the things that are produced by these machines called reads still lack information about their order and location. So the machines we have currently, they do not read your entire DNA, but rather they read just random segments of your DNA. And these segments could be short, could be long, but they do not cover the entire reference genome of human, for example. And they don't have any information about their order and the original DNA, so that we need to do that ourselves. We need to match these pieces together and then link them and build back the reference gene. How we do that? We have two types of file formats that are used for this purpose. The first one, we call it FASTA file, where we have 2 lines in this file. So the first line is this, what we call a header. The second line is the genetic content of the DNA. So you can see a lot of ACGTs, and this entire line represents your DNA. So this is the complete reference genome. And we call this format as FASTA. And this header represents the name that we use, or ID, for that sample. The other file has 4 lines per segment. The first line is the ID, similar to FASTA file. And this file, we call it FASTQ. The second line is the read itself, which is the output of the sequencer or the sequencing machine. And this is part of your DNA. So you can see ACGTs plus you can see some N. So this N means the machine could not identify whether this character is A or C or G or T. That's why we have it as ambiguous character, call it N. The third line is just a sign. You can ignore it. And the fourth one, we have the quality score. This is an ASCII code. So each character here represents the confidence that the sequencing machine has when it converts this into T. So whether it has high confidence or low confidence, that should affect the way you read or interpret these data. So these 4 lines for every segment. So if you have 1,000 of these short segments, the second line, then you will have 1,000 multiplied by 4, then you'll have 4,000 lines in this FASTQ file. And our goal next is to just map the things we have in FASTQ file to the things we have in FASTA file. The FASTA file is the full picture. FASTQ is just a few segments of your DNA, and then we solve it as if you have a puzzle where you try to stitch these pieces together and build back your complete reference gene. And this step, what we call as a read mapping, because each small piece of these, we call them a read, and then we really map the reads or these small segments to the reference, such that we do read mapping. Okay, so what's the problem about read mapping? In read mapping, this is the performance of a single sequencing machine where it can read 48 human samples in about 2 days. So it can give you a 48 FASTA file, or sorry, FASTQ file in about 2 days at 30x coverage. But when we go to the computer side or the computation, we'd like to analyze these and do the read mapping. Only we can process single human genome, which means single FASTQ file in 32 CPU hours. So you can see the bottleneck. Of course, you could use many cores or several threads to perform parallelization, but that's not the case in most of the genomic centers. They don't have cluster or cloud computing, and that will be a bottleneck in processing a huge amount of data. And read mapping is basically 71% of the execution time, and there we have 29% for other processes. Okay, so that is about genome analysis and how we get our read map to the reference genome. But what is intelligent genome analysis? So when we talk about intelligence here, we really want something very fast. So imagine if we want to do genome analysis at scale, especially in the COVID-19 time, where we have a huge amount of person would like to get their DNA analyzed or their sample analyzed to know if there's COVID-19 or not. So you would like to really do this at scale and very fast speed. And think about when we have an architecture or today's computer architecture where we move the data from the SSD or from storage all the way to the main memory, then to several levels of caches, all the way to the CPU to process, to do some computation on the genetic data. So think about if these computation are really bitwise operations. And there are several proposals that suggest to do bitwise operation inside the DRAM, for example. You don't need to move the data from DRAM all the way to caches, all the way to CPU. You could perform the computation in DRAM, or maybe in storage, or maybe in somewhere else, an FPGA chip or in GPU. That will provide great benefits in terms of energy, efficiency, and latency. The DNA is still a valuable asset. So if you hand your DNA to someone else, he might infer more information than you would ever think about it. So he can know about your disease history or the disease that you might get or your kids might have in the future or your family member are having currently. So this is really dangerous to reveal these genetic data, for example, to the cloud computing infrastructure to do processing there. So you'd like something in hand that you can do yourself the processing without the need to share it with someone else. As we say, we would like to have fast genome analysis, and fast is not enough. We want it to be scalable. So if I have 40 cores, will it scale with the increased number of cores, for example, or just remain constant after some time? So scalability here is very important. And definitely, you don't want to mess with the DNA analysis. You don't want to say that you have this disease and the person does not have it in reality. So the accuracy here is very important. We don't want to compromise the accuracy to gain speed up. And here are the different, this is today's computer architecture where we have the data from sequencing machine, we move it to SSD, to the main memory, all the way to the CPU. And single memory requests, so I'd like to fetch the data from the main memory, for example, can consume more than at least 160x more energy compared to just performing complex add operation. So always accessing memory is very expensive, plus moving the data over this power-hungry buses from one component to another component of the computing stack. And for that, we would like to, especially part of the project that we suggest in this course, try to address this issue by not moving the data and unnecessarily movement between these components. So you could directly process an FPGA chip or inside the SSD and so on. And the open question remains how and where to enable fast, accurate, cheap, and privacy-preserving and exabyte scale analysis of genomic data. As we know, most speed up comes from parallelism enabled by both novel architectures and algorithm. Building algorithms alone is not enough. Building new architecture is not enough. So we have to really make hardware-aware algorithms as well as a novel architecture that can implement such new algorithms efficiently, definitely. For more information, we already shared this learning materials, but I would really suggest again and emphasize that you need to watch the entire lecture just to get more insights about every basics that we may mention in this course or during the projects. So yeah, these are more references or the papers that we have in our research group. You could have more, you could just check and see if you get more insights or more things that you, attract your curiosity, for example, and you'd like to discover. So I'm happy to answer any question about these papers or the topic in general. Probably we can have, we can have 2 minutes. We can have 2 minutes for questions if you have. Okay, great. So yeah, at the end of the course, we will offer you some position if you are would like to continue doing the research projects with us. So we are happy to have you doing any level of research with us. Could be semester project or thesis project, master thesis, and so on, or even joining us as a full-time researcher. We are happy to have the student from ETH as well as from other places. But if you are from ETH, please send us email directly, and then we will guide you what to do next. That's all for this overview. And next, we will go in a closed session discussing the projects that we are proposing, and we can discuss that in detail, basically. All right. Yeah, thanks so much.